This is a podcast from the Nuffield Department of Medicine. Today we speak with Professor Skirmantas Kriaucionis about his research on epigenetic modifications and cancer. Hello.
Hello.
What is an epigenetic modification?
Epigenetic modification is a modification in the genome which is beyond the DNA sequence. Let me just give you an example. We have many cells in our body. They look different, they perform different function, but they have the same genome. How do they do that? They actually express different set of genes. And when I say express, I mean they use different set of genes. Some genes are being used in brain cells, others are being used in liver cells. And this process is very important to be regulated exactly and precisely. And moreover, when cells divide, when liver cell divides, daughter cell is as well looks like a mother cell. The same is for many other cell types. Therefore, epigenetic modifications are the modifications which ensure the gene expression profiles and the heritability of it when cells divide.
Can you give us an example of an epigenetic modification in cancer?
So we know that cancer is a genetic disease. Genes get mutated and this causes cancer. But what was very interesting in the last 10 to 20 years to notice that tumors have a lot of epigenetic abnormalities. So the genomes are different, but as well the epigenetic modifications are very different. So one of the most studied epigenetic modification is one which is localized on the DNA itself. It is a chemical modification called methylation. And methylation can be very different between normal and tumor cells.
Can you tell us about your current research?
We have a number of very interesting projects in the laboratory. One is to understand how gene expression is being influenced by DNA modifications. So we study proteins which are able to bind to modify DNA. We study how gene expression is being regulated. And this helps us to understand how cancer cells evolve these differences which can lead to abnormal phenotypes. Second direction is to understand how these epigenetic alterations affect cancer risk. And this is a new direction which we started to invest a lot of time recently. I'm very hopeful that we'll have some exciting results very soon.
What are the most important lines of research that developed in the past 5 or 10 years?
Some of the most important lines of the research, I think, are related to technology development. So first, human genome was sequenced, and now sequencing technology is developed so rapidly that we can sequence human genome in days. This opens a lot of interesting opportunities for us. We study epigenetic modifications, and the same tools, the same sequencing technologies actually can be used to study epigenetic modifications as well. So now, in a matter of days, we can have access to the whole modification repertoire in normal human or in cancer. So this is very exciting technology which opened a lot of different windows for us, and we are able to use those opportunities to study function of cells and epigenome.
Why does your line of research matter? Why should we put money into it?
We study cancer, and cancer is one of the common causes of distress and death in humans. Although we are experts in molecular biology, we are always looking for opportunities to discover some targets which could be used for cancer therapy. And therefore, we think it's a good and important direction to go and invest money. Although science is very unpredictable, so frankly we don't know where the most exciting discoveries will come from. But our track record of good research and important discoveries shows that we can produce some interesting and valuable insights into molecular biology and epigenetics.
How does your research fit into translational medicine within the department?
One of our recent projects actually fits it very well. When working on the recycling of epigenetically modified nucleosides, we discovered that cancer cells utilize slightly different mode of recycling of these epigenetically modified nucleosides. And we can use this mode to interfere with cancer growth by killing cancer cells. So we are very excited about this direction. We are looking at cancers which have this altered ability to recycle modified bases, and one of those cancers is actually pancreatic cancer, which is very aggressive and difficult to treat cancer. And we are now very strong to pursue translational opportunities to see whether or not our insights can be used to develop valuable and useful therapies.
Thank you very much.
Thank you.